07530 905 152 for your mobile massage therapist.
Across the World Wide Web, on your mobile devices, on social media platforms, this is Jambo Radio! www.jamboradio.co.uk. We are multilingual. We are conversational in the languages you best understand. We are Jambo Radio! Jambo Radio, inspiring transformation.
This is the Science Show with Dr. Dada. It's just so nice, you cannot do without. This is the Science Show with Dr. Dada. It's just so nice, you cannot do without.
Well, hello, and welcome to the Science Show on Jambo Radio Scotland. And this is Dr. Dada welcoming you. And today we're going to be exploring a very interesting topic, very, very encouraging. Today's guest is Professor Andrew Jamieson, and he is leading the charge in the fight against one of humanity's most urgent health challenges, and that is drug-resistant malaria. Now, malaria, believe it or not, is a disease that still claims hundreds of thousands of lives each year, and it has become harder to treat as the parasite develops resistance to current drugs, and that threatens decades of progress in global health. Now, Professor Jamieson's groundbreaking research focuses on designing innovative chemical solutions to outsmart the parasite, if you like. And he also does many more things, as we'll find out in today's interview. Now, his team has developed a novel class of compounds that show immense promise in overcoming this resistance. So the question for the week is very, very interesting. Could this be the end of drug-resistant malaria? Well, let's welcome Professor Andrew Jamieson to our studio. Welcome, Andrew.
Good afternoon.
Yes, it's
Pleasure
really good to have you with us today.
Okay, thanks very much. Pleasure to be here. Tell you about some of the research we've been doing at the University of Glasgow.
Very good, fantastic. And fittingly, our first highlight for today's show, first science news highlight, is really about that research. But before we go into that, let's talk a little bit more about Professor Jamieson. So Professor Andrew Jamieson is a professor of chemical biology at the University of Glasgow, where he leads a cutting-edge research group. And his research really is into peptide and peptidomimetic synthesis. Yep. Did I say that right?
Peptomimetics, yeah.
Peptomimetics.
Yep.
Peptomimetics.
Where my research group enjoys saying it as much as you do.
Great. That's great. And this is really to help address critical, health challenges like cancer, Alzheimer's disease, and of course today we're going to be focusing on malaria. Now, Professor Jamieson, would you like to tell us? We'd like to get to know about you a little bit. Were you born in Glasgow? Are you from here?
I was, yeah. I'm a dyed-in-the-wool Glaswegian.
Great.
I was born in the Royal Infirmary in Glasgow. I was brought up in a little village just out of Glasgow called Straven. It's where I live now.
I see.
I did my degree, my PhD at Glasgow. Yeah, I even got married in the University Chapel.
Wow.
So, yeah.
That's fantastic. You're a true Glaswegian,
yeah.
A true Team U of G.
Yeah.
That's amazing. That's really nice to know. But I know you've also held research positions across the globe, including postdoctoral fellowships. Perhaps could you tell us about where you've been?
Yeah, so when I finished my undergraduate degree, I stayed at Glasgow to complete my PhD in synthetic chemistry, and I became interested in a particular type of molecules and how to make those. And one of the world leaders in that field was in Montreal. So I moved to Montreal in deepest, darkest January, thinking that would be okay. I very quickly learned about cold weather that's worse in Scotland. So I spent a year and a half in Montreal working on that project, trying to develop a treatment for preterm labour. And then I got interested in a slightly different type of molecule, and so I moved down to the United States and joined the research group of a man called Andrew Hamilton. He was a provost at Yale University at the time. And during that period, he was headhunted to come back to the UK. He's a Scottish guy as well. He came back to the UK to be the Vice-Chancellor of Oxford. And so I came with him as his senior fellow to set up the lab in Oxford. I spent a year there doing some really interesting science. And then I set up my own academic group in 2010 at the University of Leicester in the Midlands of England.
Nice. That's a fantastic track, you know, from Glasgow to the US, then back to England, and then now back to Glasgow. So from Leicester to Glasgow, what happened there? How did that happen?
So it was after the kind of financial crash of 2008. So there weren't a lot of universities recruiting, and a job came up at Leicester. And actually, I didn't even know where Leicester was. So in the train on the way through to the interview, I had to look up where Leicester was, where I was travelling to. And I was really impressed by what they had going on there in terms of their science departments, chemistry, biology, and physics. And so I moved there thinking I'd spend a few years there and then I'd hopefully get the opportunity to move home. But that was a period when Leicester City were doing really well at football. The local rugby team, Leicester Tigers, were doing really. I was having a fantastic time, both at work. I got married to my fiancée, and she moved down to Leicester. We were just having such a great time. We stayed with 2 kids down there, and then the opportunity came up to move back to Glasgow. They had a position, and so they approached me to apply for that position, and then that all worked out, and I moved back to Glasgow in 2016.
That's nice. That's really, really nice. It's nice to have you back. It's really good, isn't it?
It's good to be back.
Yes. And I know you're also is that correct, that you're also the founding director and CSO of Keltic Pharma?
Yeah, that's correct.
Tell us about
that. Our most recent endeavour. So yeah, this is off the back of the malaria research that we did initially.
I see.
We were approached by the European Union Malaria Fund, who saw we published our initial research in the journal Science, which is really kind of high profile. Some of your readers may read the journal.
Yeah, Science is a big one.
Yeah, so we're approached and they said, you know, this is fantastic. We would really like to be able to fund you to take this project forward and try and develop a drug for the treatment of malaria. And so we did. That all worked out. Keltic Pharma was born. And we've kind of grown since then. We've now got 13 employees in the lab, predominantly scientists.
I see.
Doing chemistry, biology, pharmacology.
Interesting. Yeah. Is it based in Glasgow?
Yeah, so we're still technically a spin-out. So the employees are housed within our research labs at the Advanced Research Centre, the ARC, which is our new fancy building.
That's really nice. It's very convenient as well. So you've got your group based in the ARC as well.
Yeah.
And so you can have this industry, academia interaction.
Yeah, I mean, it's a real kind of strategic advantage for the company because the people in the company can learn from the academic group and vice versa.
Yeah.
Because I think research within an academic context is quite different to an industrial context.
Yeah.
In academia, you go after what's interesting, but in industry, at the end of the day, you need to produce a product.
Yes, absolutely. And the focus in terms of output, as you've mentioned, is different as well.
Yeah.
Academics love to publish, quite quickly what you find. They're so excited to share. But it's slightly different in industry, isn't it? Very nice. So it was really interesting for me when I saw the press release about your research. I saw it earlier this month. New drug could help fight against treatment-resistant malaria, right?
Yeah.
That sounds so wow. Oh, okay, that's very interesting. It's nice to see what goes in, as in what approach is new with this new sort of drug, right? And this is published work. If you'd like to find out more about this research, you can read about it in ACS. So it's published in the Journal of Medical Chemistry. Is that right?
Yeah, medicinal chemistry, yeah.
Yeah, medicinal chemistry. And the research was supported by the EPSRC, Biotechnology and Biological Sciences Research Council, which is BBSRC, and the Bill and Melinda Gates Foundation, BMGF, as well.
Correct. Yeah, good.
That's interesting, isn't it?
Yeah.
I was quite surprised when I saw EPSRC on there. I was like, oh, is that within the remit of EPSRC? But then when I looked into it a little bit, as we'll delve into as well, I realized that, wow, this is really, there's a lot of chemistry actually going into this. It's not just, dealing with the biology aspect of things. So we'll learn more about that. But the Bill and Melinda Gates Foundation, so is that do you apply to them directly for funding as well? How does it work with that sort of funding?
Yeah, so that funding came about. So my colleague, Andrew Tobin, who's kind of the biologist where I'm the chemist, he spoke at a conference. He was approached by someone from the Bill and Melinda Gates Foundation. Same idea.
Wow.
We think this is a great idea. We'd be interested in funding your research. And so we wrote up a proposal and we sent that to them, outlining what we would do if they were to give us funding, and that worked out. So they've now given us 2 grants.
Wow.
So yeah, it's really funded a large part of the kind of industrial program to develop a treatment for malaria.
Wow, that's amazing. Do you know if they fund physics related things?
So interestingly, they do. They fund anyone who's got a kind of technology that could transform, and it's transferable to different areas.
Yeah.
That can help global good. Then would be interested in hearing. Yeah.
That's amazing to know. So many funding opportunities, isn't it? So yeah, PSRC is on there. BBSRC is there as well. Yeah. It's really good to see. Shout out to all these funders. They help make these developments possible. So you grew up in. Could you repeat where it was that you grew up?
In Straven.
Straven
is the village. Yeah.
I'm suspecting it's spelled slightly differently.
It's spelled Strathhaven.
Right. I thought Straven sounds a bit different from what I saw. So Straven, that's interesting. It's a bit like Leicester as well, isn't it? So you've been connected with these places that test your understanding of UK pronunciations. So what was it like growing up in Straven?
It was great. I mean, I've moved back there for kind of the same reason. It's a great place to raise kids. It's a really safe village. We have a fantastic park, Straven Park. It wins lots of awards every year. So yeah, it's a great place. The high school's very good. The primary schools are very good. I had a great time growing up there. I had 2 older brothers, so you sort of run about as a kid climbing trees and with them. Primary school was wonderful. My kids, there are 2 main primary schools in Straven. I went to one, and my kids actually go to the other, and there's a little bit of rivalry and play football together and stuff. So it's really good, and the high school's a fantastic place. My kids, my eldest son has just started high school there. And it's completely changed. So the building that I went to is not there anymore. The school's been completely rebuilt. I see. But it still is an exceptional high school.
Yeah, and your track record is testament to the fact that the schools must be doing something right.
Well, it was where I first got into biology, actually. That was where we had a teacher, Miss Rogers. She was fantastic. And I just fell in love with biology at the time. And I actually went to university, to Glasgow, to study biochemistry. That was what I was going to do. And then going through university, I enjoyed the chemistry labs more than the biology labs. It's interesting, the demonstrators who help you with your experiments. In the chemistry labs, they all seem to be having really good fun. And the biology labs,
blowing things up.
Yeah. So literally, we used to do a reaction called the thermite reaction, which is literally creating an explosion. I thought, this is really good fun. I want to do this. I took things from there.
Wow, that's amazing. It's so nice to see what ignited your interest in chemistry. And what was it like adapting then to so going a little bit further in your career? What was it like adapting to the influences of different environments when you went to Oxford, you went to the US? What was that like for you?
Yeah, it was good. I mean, each institution I went to was very different. So Montreal. So I went to the University of Montreal, which is the French-speaking university.
Wow.
Speaking no French. I mean, it was crazy. We went in, and it took a little bit of time to kind of integrate with the Québécois, the local people within the lab. But I think they have a similar mindset to people in Scotland. And so we got on really well, and they kind of tolerated my inability to speak French. And we ended up doing things like going snowboarding and things like that. And it was really good. I felt like I kind of integrated well there. Yale was a very different experience. So working for the Vice Chancellor, there's a bit of prestige about that.
Nice.
So I mean, when you're a research fellow, you spend most of your time in the research labs. But we did get the opportunity to go and see seminars from some of the top scientists in the world, Nobel Prize winners and the like, which was an outstanding experience. And similar experience in Oxford. There was a kind of conveyor belt of seminar speakers that are the most high-profile scientists in the world come round. You get to hear.
Imagine.
I remember
Yale is very prestigious indeed.
It was an outstanding experience. Probably my favourite part of that actually was playing for the rugby team.
I see. I was coming to that. So I know you're an avid runner, rugby player, and snowboarder. Now these are like nice hobbies that provide an outlet to your, different aspects, other aspects of life. So would you like to tell us about that?
Yeah, sure. So I took up snowboarding during my PhD. It was the indoor snow slope in Glasgow.
Yeah.
And then when I moved to Montreal, they have lots of skiing facilities kind of round about the city. And so we go every week during the winter. It was one of the ways I actually got to know the Québécois people within the lab.
I see.
Running is great. It's a great release to go out running. I've got a torn meniscus at the moment, so my running's limited at the moment. But my physio's doing a great job at trying to get me back on the road. And then rugby. I've been playing rugby since I was 8 years old. Played all the way through school, all the way through university. I actually broke my neck at one point.
Oh no!
So that kind of ended my career for a little bit. Played again and then broke my nose playing in the States. So I kind of hung up my boots at that point. But now my son's playing rugby. I now coach his mini rugby team at Straven Rugby Club. So that's a fantastic thing to do on a Tuesday evening and a Sunday morning.
Yeah, it's nice to see the balance between such an intense, research career and personal interests like that. It's quite nice. Yeah, my thing is music. I tend to, dabble into music for fun, which is quite nice. And speaking of music, actually this next one is one of my songs that I kind of put together. So this is entitled Jeje Love, and that will be our music break, and we'll be right back after that and continue our interesting chat with Professor Jamieson. We'll be right back.
Leave me with my babe. Yeah, you know they bother anybody. Yeah, now Jeje we they move. No mind my latest groove. I know they look your way, stay smooth. Yeah, just to go your own. No put your nose where you no belong. No put your mouth for talk when no be yours. This life not soft, no fuss. Make we live without remorse. Life is sweet when you find true love incomplete. It be like joy if they give happiness where you know fit peace. You know don't need any other gist. Love go chase away the twist. It go fill your heart with peace. We build our one, no stress. Everything up your finesse. No wahala, no distress. Just good vibes with the bless. We no send, we just progress. Who want talk with the guest? My baby, now my true success. Together we dey shine, no less. So they ask you waiting show. Tell them say now love they grow. No need for noise, no need for fight. We just the glow, we dey alright. Life is sweet when you find true love, it's complete. It be like joy they live. Happiness where you know fit miss. You no go need any other gist. Love go chase away the twist. It go fill your heart with peace. Just with the live, no stress, nothing negative. With my baby by my side, we go the alright, no divide. Peace of mind within inside. All we need now love to guide. Till the end we go decline. No matter what the people say, we go follow our own way. In peace and love we go stay. And steady joy till the break of day. Life is sweet when you find true love, it's complete. You feel like joy they live. Happiness where you know fit means. You don't go need any other cheese. Love go chase away the twist. You can fill your heart with peace. I just with the live, no stress, nothing negative. With my baby by my side, we go the alright, no divide. Peace of mind within inside. All we need now love to guide. Till the end we go decline.
Yes, that was JJ Love, and this is the Science Show with Dr. Dada on Jambo Radio Scotland, broadcasting from the city of Glasgow. And we've been talking today about the end, potentially, of drug-resistant malaria. And today's guest, of course, Professor Andrew Jamieson, has been talking to us about that. And we'll get more into that in a moment. But before that, we're going to just highlight one other highlight in the news when it comes to the world of science. And this is something that's quite close to my heart. And it's to do with the subject of light. So scientists have discovered that under certain conditions, a laser beam can act like an opaque object and cast a shadow. And that's quite mind-blowing when you hear, for the first time, you're like, what? Photons don't interact. There's no way that is possible, right? Well, this surprising finding is something that could lead to new ways of controlling light, such as using one laser beam as a switch for another. So if that's on, then the other one's blocked, that sort of thing. Now, this challenges the traditional understanding of shadows on how that works, and it opens up new possibilities for a range of technologies. But before you get too upset, if you really look into the paper, which I'm going to share in a moment, it turns out that this interaction actually needs a medium, right? It's not in vacuum. It's not like you have one, bunch of photons blocking another bunch of photons. That would be indeed very strange. But really what they do is they use another medium, like a crystal, and they shine one kind of light through the crystal, and that changes the properties of the crystal. And that sort of makes the crystal more opaque to another wavelength of light. So it's quite an interesting one. So it's done by the researchers from the University of Ottawa, and they've shown that under these sort of special conditions, a laser beam can act like an opaque object. So it appears to be the case, but actually it's the medium that's transferring it. And I can see how this could be a little bit controversial depending on how you frame the headline, But yeah, this work is published in Optica, and the title is Shadow of a Laser Beam, published in Optica number 11. And you can find that quite easily. The last author is J.S. Lundin. So yeah, that's something that caught my eye in the news in recent days. Now, let's come back now to the topic of drug-resistant malaria. Our question for the week was, could this be the end of drug-resistant malaria? And of course, that's talking about the discovery made by the group of Professor Jamieson. So, Professor Jamieson, we're really excited to talk to you now more in depth about this fantastic, potentially groundbreaking work on this new drug for malaria. So for our listeners who might not be familiar, Could you start by explaining a little bit why malaria is such a persistent global challenge?
Yeah, sure. So there's still hundreds of thousands of people affected by malaria every year. 200 million, 600,000 people die of malaria every year. One of the scary parts of the statistic is that 75% of the people who die are under the age of 5 and pregnant women, which makes it a particularly devastating disease. So parasites that live within mosquitoes are what cause malaria, and they're highly adapted organisms that have a kind of pool of proteins that if you apply drugs to them and that you don't apply enough of the drug to kill the parasite, then the parasite can then start to develop resistance. They can evolve essentially to overcome the drug. And then as that parasite's then transferred to somebody else, the drugs are no longer effective.
Wow. That's scary, isn't it? And that just really highlights that it is a global health emergency, and it's a really, really important area for research, isn't it? Otherwise, all those people dying, the new drugs are so desperately needed. Now, talking about the development of drug-resistant malaria, obviously that's a major obstacle when it comes to controlling a disease. If the disease becomes smarter and said, "Oh, right, you think you think you're cool, you're smart, you develop resistance against that solution," and then you develop another one, it develops resistance against that. It feels like very quickly you might run out of options. So could you explain how this drug resistance develops? The parasite itself, how does it know to develop resistance?
Yeah, so this is, I guess, something that's, I think, not really well understood generally. So parasites have, proteins, the parts of the parasite that allow it to be a parasite. And within that parasite, there are kind of versions of those proteins that are broken, essentially. They're not quite produced correctly, but they still work. They're not quite as effective as the way they're supposed to be built and the way they're supposed to work. But they're kind of, we call them mutants. And what happens is the drugs hit the targets that are normal, and those mutants, the drugs don't affect. And so the parasite is able to survive because although those mutants don't work as well as the normal proteins, they can still work. And that allows the parasite to survive. The drug gets washed out of the parasite, and then the parasite survives. And what happens is they produce more of this mutant protein that allows it to survive, and the drug all of a sudden no longer works.
Wow, that's a really nice explanation. Thanks very much for that. So, it's a bit nightmarish, isn't it?
It is a little.
It's like they're little villains, these parasites. Anyway, so now your research uses a new approach to then fighting this malaria. Can you tell us about that? So if I understand correctly, it borrows a strategy from cancer treatment, doesn't it? Yeah,
it does. So I guess to kind of follow on to the mutant analogy. So parasites have these mutants, and that's how we get round a particular drug, because a particular drug will target a particular protein, generally speaking. And if all drugs are developed to target that same protein, once your mutation is formed and the parasite can survive, it doesn't matter if you tweak the drug, it's not going to work. So what we need is new drugs that work by a completely different mechanism, meaning they're binding to different proteins.
I see.
And so if we can do that, what we can do is use what are called combination therapies. So using more than one drug in a single capsule, a single dose. And so what happens is, you're hitting one target, it's trying to evolve, but you're killing most of the parasite, and then you're hitting another target and it's trying to evolve. But the combination of both allows you to kill the drug, to kill the parasite, without that resistance having a chance to develop that the parasites aren't able to overcome. So the more drugs you use, the better. The more drugs you use that have different mechanisms, so hit different targets, the better. And so that's the front line at the moment. So we use what are called artemisinin and combined therapies. So artemisinin is the main drug that we use against malaria, and it's used in combination with these other drugs. Yeah. The problem is there's resistance developing now to artemisinin and to these other drugs, and so we're running out of options. And so what do? So we develop new drugs that work by different mechanisms, and then again we can put together combinations of these drugs and use this to treat the malarial parasites. Will we ever overcome resistance? I think parasites will always develop resistance. The question is, can we eradicate malaria? So can we develop enough drugs and distribute them effectively enough and use them in combination with vaccines and with insecticide sprayed bed nets? That combined approach, can we use that effectively to eradicate malaria? The answer to that is yes.
Good.
Is there the will? Is there the funding to do it? That's the barrier.
I see. That's interesting. So the answer is kind of yes and no then. Could this be the end? It's yes and no. Depends on what you mean by that. So I think you could say if you could eradicate malaria, then you would have,
You would have done that,
Yeah. So you can say yes to the question.
Resistance,
Good. So can you explain? So I'm going to, just a random question here. There's a terminology that's kind of bugging me, and I'd like to get you to explain it. So can you explain what a covalent kinase inhibitor is? Is it kinase or kinase?
Kinase.
Kinase.
Yeah, we pronounce it.
So, and how does it differ from previous types of malaria drugs?
Yep. So if you have your protein target, basically your drug, what it does is sticks to that protein. And you imagine a spanner in the works, right?
Yeah.
That protein's got a job to do. And it can't do it when that drug's stuck to it.
Yeah.
Okay. So that's what normally happens with a standard non-covalent drug, as we call them.
Oh, I see.
And so what happens is eventually that drug will unstick. It will come off. Okay. And then the body can just destroy it and it goes away. And the effect, we lose the effect. With a covalent inhibitor, what happens is it sticks, and then it has the equivalent to like a grappling hook that sinks into the protein, and then that drug won't come back off. It's stuck there forever. It's covalently bound. And that's the difference between a covalent drug and a non-covalent drug. I
see. Wow. So it's all in the binding.
Yep.
Wow, that's amazing. And is there something to do with, does anything happen at the sort of RNA level at all in this whole picture?
Yeah, so the target that we're going after is an enzyme, so it's a protein that does work.
Okay.
And that enzyme, we believe, is involved in a biochemical process called RNA splicing.
Okay.
So it's kind of an interesting one, right? So in your cells, you have your nucleus.
Yep.
In the nucleus, you have all your DNA.
Yes.
Okay. So the analogy I make is that's kind of like your granny's cookbook, right? So there are lots of recipes, right? But just like your granny, she doesn't cook all of the recipes, right?
Yep.
My favourite's the scones, okay? Within a cell, the cells don't use all of the DNA to describe or make all of the proteins that are coded in that DNA.
Yeah.
Okay. So one of the ways that it can make proteins, and it can actually change what it does, right? So in Granny's cookbook, she never really follows the recipe fully. There's pens, she changes the amounts and stuff like that. So that's a process in cells called RNA splicing. So you take the code and you cut bits out.
I see.
And what you get is slightly different to the DNA code.
Yeah, I see.
So that's the RNA splicing process. So our target, which is called PFCLK3, is involved in that process of RNA splicing.
Ah, that's interesting. And that's how it kills the parasite. That's how it's such an important target.
Yeah. So cells can't survive unless they can produce proteins.
Yeah.
And so cells have life cycles, okay? They do different things at different times. So most human cells, for instance, will last a day. And they do different things at different times.
Yeah.
But all of those processes, you need that code to make proteins to do the jobs that are required at that particular time. So if you put a spanner in the works of the process to produce the proteins, then it works all the time. And that was really the big breakthrough with this particular target, that it would appear that it's able to kill the parasite at all life stages.
Wow, that's to me. I mean, hearing that, I'm like, how is it ever going to develop resistance to that?
Well, the idea is that you'll kill the parasites and
Yeah.
will eradicate the parasites
Yeah.
and remove it as a problem. And then, well, resistance goes away as a problem.
Yeah. So then it's more optimistic than I thought from what you said previously. It seems actually this is because I mean I can't think of a way that, or can you think of a way that it develops resistance to that, something so different?
So with everything in science, the thing to fear is what you don't know.
Yeah.
And we don't know what we don't know.
Yeah.
So if we use a covalent inhibitor, it's able
Yeah.
to kill the parasite at all life stages. Mother Nature has a nasty habit of teaching us. And so we don't know how the parasite could evolve resistance yet. So this is kind of the next stage of our drug development is when we apply the drug. So all of the tests that we do so far, there's kind of a high throughput one that we do fairly regularly, which looks at the pool of mutants and whether the parasite has mutants there that could overcome the drug. And it looks like it doesn't. And then there's the evolved mutants, when you put the parasite under pressure, so drug, can it evolve over time? Can it change its genetic code in order to overcome the drug? That takes longer, but again, it looks as if it can't do that particularly effectively.
I see. I like that then. I mean, so you're cautiously optimistic.
Absolutely.
But it's a really good position to be in.
Yeah.
When you can't come up with a way for it to actually develop
we're always hunting the next problem.
Yeah, exactly.
And this project is very much a case of that.
That's fantastic. It's a great project. We're going to talk more about it after the next music break. And for this, we'll be listening to Tenny Wait. We'll be right back.
Dr. Dada Entertainment.
Jason.
I can't take your pain away, but I can make you feel better. If you stay another day, we can do it together. From sundown to sunup, we go work the work together. We go do the do forever. From sundown to sunup, we go work the work together. We go do the do together. Yeah. Baby, wait for me. Yeah. Would you stay with me? Yeah. No regrets, baby. Like a soldier, would you fight for me? Baby, wait for me. Would you stay with me? Yeah. No regrets, baby. Like a soldier, would you fight for me? Cause I'm always on time. In the night, in the day, to give you my life. My love is yours. Give it to you, no be joke. Baby, now you that for sure. It took up. My love is yours. Give it to you, no be joke. Baby, now you that for sure. One lay bow. One lay, one remove. You want the color come here. Make we stop the fight, oh no. One lay bow. One lay, one remove. You want the color come here. Make we stop the fight, oh no. Bring it back for me. I can't take your pain away, but I can make you feel better. If you stay another day, we can do it together. From sundown to sunup, we go work the work together. We go do the do forever. From sundown to sunup, we go work the work together.
Wow, that was Wait by Tenny, and this is the Science Show with Dr. Dada on Jambo Radio Scotland, broadcasting from the city of Glasgow. I would like to take a moment at this point to shout out to our sponsors. We'd like to say thank you to IOP Scotland and to the EPSRC through the University of Glasgow. And we'd like to say give a shout out to all the people in the studio as well with us. We have Baron Umoren, or Otuekong Umoren, who just passed his PhD as well. So congrats, Dr. Baron. It's nice to welcome you to the club. And of course, again, another reminder that you can listen to this show in very many different ways. You can listen on DAB, so we're on air on DAB. So whether you're in the car, whether you're out in the garden, if you have a digital radio, you can listen to this show live. And you can also watch us live and listen live at jamboradio.co.uk and also through our YouTube channel. So Jambo Radio Scotland on YouTube. So please remember that. And also, in our discussion, so of course, the question for the week that we're looking at today is, could this be the end of drug-resistant malaria? That's a very, very interesting question. And the answer so far I'm getting is, it seems to be yes, but we'll learn a little bit more about what's involved with that. Well, to join the discussion, please feel free to call us. And the number to call is 0141 530 2787. Once again, that is 0141 530 2787. If you have any questions, or if you just want to, weigh in with your thoughts on the question for the week, you'll be welcome to do that. Now, let's carry on our discussion about this whole idea of drug resistance and development of this new drug that, is really, really promising when it comes to breaking the resistance, as it were, So now, when it comes to determining how the parasite reacts to the drug, as to whether it actually is able to develop resistance or not, what kind of tests do you need to do that gives you the confidence that, okay, I think we've got something good here?
Yeah, so when we're developing drugs, we're constantly making molecules in the lab. And we put those through a series of tests.
Yes.
So we start with the kind of easy ones of, does it attach to the protein and stop the enzyme from working? And then we move to parasites.
Yep.
And we see if it kills the parasites, and it's kind of a yes, no, they are dead or they are alive type assay, which is great. And then we move on to more advanced assays, and we tend to collaborate with people internationally. Nice. So there's a lab, the FIDIC lab, that we collaborate with, and what they do is they feed the parasite lower levels of the drug or higher levels of the drug, depending on what the experiment is, and they look at whether the parasite dies off, and then whether it comes back again. So whether there's very, very small numbers of parasites that have somehow been able to dodge the effect, if you like, of the drug, in which case they will replicate and then come back, and that's called a recrudescence test.
I see. Reconnaissance.
Recrudescence.
Recrudescence.
Yeah. This was a new one for me until fairly recently, the recrudescence assay. Yeah.
Wow, that's interesting.
It's a lovely word.
Yeah, it is. It sounds good. There's so many things that go in, isn't it, into actually being able to, the road to getting it out to patients.
Yeah.
It's quite a long one, isn't it?
Yeah.
But so far, what would you say have been the biggest challenges, some of the biggest challenges you faced in developing this new drug so far?
So I guess we go back to the start. So I think identifying a new target, a new way to kill the parasite, and this was really an innovation by my collaborator, Professor Andrew Tobin. He was the person who identified this kinase that's essential. If you are able to inhibit that protein, stop it from working, the parasite will die.
Wow.
Which was some fantastic work.
Yes, indeed.
And then he partnered with us to, we make the molecules, and we can design and tweak them to get them to do what we want them to do. So that initial discovery, that was huge. we screened a whole load of molecules. So we partnered with a company, GlaxoSmithKline, and they have a facility in Spain called the Tres Cantos Screening Facility.
I see.
And so we had an assay, and a postdoc went to Spain and took their hundreds of thousands of compounds and screened them all against the kinase, the enzyme, and we discovered a molecule which we affectionately call 051.
Wow, nice.
Which is part of the code for that molecule.
I see.
So finding that molecule, that was another kind of hurrah moment, if you like. And then we were sitting around discussing, right, how are we going to take this drug? How are we going to make it better? And it was at that point, so it was a conversation with the PhD student who ultimately did the majority of the work in our.
Is that Sky?
Sky, yeah.
Shout out to Sky.
So Sky Brattel is a Scottish PhD student who has worked on this project now for the last 3 years. So she finishes her PhD in December and has been fantastic, very hardworking, inspirational ideas. It was that sitting down initially and saying, right, okay, what we're going to work with, what are we going to do with your PhD? How are we going to take this drug and make it better and overcome the problems? And said, well, okay, well, let's think about it systematically. What's the problem with it? And one of the problems we had was selectivity. So it would stick to the protein that we wanted it to stick to, but it would also stick to others.
So, I see.
And so Sky went away and did some really fantastic research on looking at the sequence, the makeup of the proteins. And she simply looked for differences. And she discovered that there's a single amino acid, so proteins are made of chains of amino acids. She found a single difference between our target and all of the other targets, bar 2. And there are hundreds of these in the human body. Yeah. And she said, if we can use that difference, then we should be able to make a drug which is selective. And if you make a selective drug, you don't get the off targets, you don't get the side effects. Yeah. And so that was we had identified what the problem was. We had come up with a way to overcome the problem. And then it was just a case of, right, now we need to design the molecules. Yeah. So we used lots of advanced methods to do that, lots of really interesting two-dimensional software. We're now using virtual reality. So before we would use a computer screen, you would look at the protein, which just looks like a big blob, really. Yeah. But we can now use VR.
And you can kind of rotate the molecules.
It's fantastic. You can go inside the protein and you can see what it really looks like. And you can say, right, okay, well, there's like a little pocket there. Let's put something in that pocket. and that's basically the design process.
That's fantastic.
You design the molecule, go into the lab and make it. And that's what Sky was inspirational in our kind of ability to design those molecules, go into the lab and actually chemically synthesize them. And then we tested them and it worked, that was the next.
That's the next eureka moment.
Yeah, like, sometimes you have these ideas, right, in science, and you go, I wonder where this is. Let's test it.
Let's check it out.
You test it, and when it works, you're like, wow, that's amazing.
Yeah, it's one of the things I love about doing science, which is there are parts of it that are really difficult and challenging, but those moments where you get these revelations, if you like, or these discoveries, it's just something, isn't it?
Yeah,
so it really makes it worth it.
We talk about hitting your head off a brick wall. Yeah. So in science, but 99%, particularly in chemistry, 99% of what you do won't work, and it's like hitting your head against a brick wall. It's really painful. It's an emotional roller coaster. Yeah. And then sometimes you just do that reaction. It works. You make the molecule you want to make. And it's like knocking down the wall, and it's just that that feeling that's what drives us to then move on to the next wall.
Yeah, absolutely.
Fantastic process.
It is indeed, and of course, taking this, that process, extending it to the point where it gets to the patient, it's quite a long road, isn't it? It takes sometimes even years, doesn't it? So, how close are we to seeing the practical applications of that drug or this discovery in the real world, would you say?
Yeah, so there's various different stages of the drug discovery process. We're probably about halfway through. So we're working on this probably about 5 years in total now. I see. Nice. So the next stage is called lead optimization. So we take what we've got, so the next thing we need to do is find out about toxicity in the human body. And then we would partner with a larger pharmaceutical company to take the compound into clinical trials.
I see.
And actually, the reason that takes most time is the kind of bureaucracy, is the red tape, the raising the funding, because it's extremely expensive to do clinical trials, and finding investors, because it's very high risk. Lots of drugs fail in clinical trials. Either when you when you do all your experiments in the lab, they work. But when you put them in humans, they just don't work.
It's just so many other things to deal with.
So that process of planning, raising the money, and before you even get to the start of the clinical trial can take a long time.
I see. Wow. Yeah. Well, we hope it gets, especially to those areas of parts of the world hardest hit by malaria. We hope this kind of progresses quite quickly, and they can really feel the benefits of this research. Now, many diseases, not just malaria, many other diseases are becoming drug resistant, aren't they? Now, do you think there's something that could be learned from your approach? Do you think this approach could kind of translate to these other diseases as well?
Yeah, absolutely. So we borrowed this approach from cancer therapeutics. So this has been applied in cancer for about a decade now, and it works. There are compounds in clinic. The problem, again, tends to be toxicity. So it works and it kills the cancer, but you get side effects. And so what we're trying to develop now is take the strategy, work out what causes those side effects, and then modify the drugs so that you don't get those side effects. If we can crack that, then absolutely. So it'd be applicable to anything where resistance could be a problem. So malaria, antimicrobial resistance is going to be one of the next big things,
absolutely.
right?
Yeah.
So this strategy absolutely could be transferred to those other clinical indications.
That's amazing. Very, very nice. Yes, that's deserving of an applause indeed, isn't it? So, I just can't get out of my mind this whole idea of virtual reality. It's so mesmerizing, isn't it? To think you're actually walking in the molecule and just looking around. So do you have that in your laboratory, in your sort of facilities in the Advanced Research Centre at the University of Glasgow?
Yeah, so we have a VR suite.
I see. Is it like glasses that you put on? Or?
Yeah, so I got into this fairly early using VR, even before the VR suite was kind of set up. So it's a set of goggles. It's Oculus Prime goggles. You buy them from kind of the local shops, and then there's special software that we use that allows us to visualize the proteins, the targets.
And can you interact with the molecules?
You do, yeah. It's it's a computer game essentially.
Wow.
For scientists.
That's great.
It's really good fun.
That's really, really amazing. I should come and visit one of these days.
Absolutely.
And have a look. Oh, wow, that's good. Well, it's, as I, probably mentioned to you, time flies. Time really flies on this show. So there's not a lot of time left. But before we conclude on the interview, what keeps you optimistic about the future of malaria treatment, and how can the public and policymakers support this fight against drug-resistant malaria?
I think the policymakers are becoming more and more aware, and I think that's like people care about what they know about, and I think keeping up the kind of publicity of ensuring that the politicians in particular know about malaria and know about the impact of malaria and other diseases like malaria. Will be incredibly important. So institutions like the World Health Organization, we're hoping to partner with Medicines for Malaria Venture. They do a lot of work to publicise the problem, and I think that's really the secret to pushing forward funding and resource to solve the problem. Yeah, very good. Thanks very much. Thanks very much for that. And that really brings us to the end of the interview. Yes, there might be a part 2. We might maybe have Sky on a future episode on the show, but there's a lot to talk about when it comes to this. It's a really exciting and fascinating research, isn't it? But anyway, that brings us to the end of our show today. And once again, welcome and thank you very much for tuning in to our show.
We're so happy to have you. Those of you listening at home, whether listening live right now or watching and catching up with the recorded version of the show. Again, you can always catch us live on DAB. So next week will be another time to do that. But anyway, with that, we'll bring our show to an end, and to sing us out, as you'd say, we'll listen to General Defao, and the song is Leto Shalom. And with that, I'll say, stay curious.